Hello students, this is Dr. Faizan Mirza. I'll be covering questions related to biological molecules in this video. Okay, so this question came in variant 2015, variant 21. They have shown us a diagram of tristearine, a triglyceride, and a phosphatidyl and a phospholipid. And they are saying that the structural difference of between the two molecules are shown in figure 5.1. The table is given to us. They want two structural differences other than the number of different atoms, types of atoms in the table. So they have mentioned length of fatty acid that are all the same and different lengths. So you have to compare these two phospholipids together. So what can we compare? We can say that obviously there is a positive charge and a negative charge here. So this is having charges. This is polar. This is non-polar. There's phosphate present, and you do not. In fact, we can mention phosphate because the type of atoms are different. We will not really account. It won't be counted as an answer. Tails, you can say number of tails. So here they have three tails, and here you have two tails. So you can see three and two tails. The number of these bonds, the ester links, the ester bonds is three. Here the ester bonds are two. So you can mention that as well. There's a double bond here. So this is a kink here. You can see not exactly, although kink is not shown to us, although they should have shown, but this can be seen. This can be mentioned as the carbon-carbon double bond is there, or this is an unsaturated fatty acid tail because of the presence of this carbon-carbon double bond. And you do not see any carbon-carbon double bond here. So any two can be mentioned from this diagram in this particular table. Further, there the question goes to how exocytosis occur. They are showing to you the secretion of amylase and trypsin into a duct. So this is the duct outside the cell. This region is the duct. This is the plasma membrane of the cell. This is the cytoplasm of the cell. Since the cytoplasm is shown on this side of the membrane, so if this side is cytoplasm, then obviously opposite side should be the duct. So this is the link here. So what's happening? The enzymes are being packaged into vesicles and exported out from these cells, that's shown in figure 5.2. So these are the vesicles, and these vesicles they are just releasing the enzymes outside the cell. These vesicles, you can see they are having phospholipid bilayer. The phospholipid bilayer is there, and they start to move towards the cell membrane. The phospholipids move, so the polar heads they start to bond together. The tails they start to bond together as well, and the vesicle starts to open up, releasing all its contents out of the cell. Now, when it is moving, it will move in that particular direction. What structure was there that gave it direction? There were microtubules here, because we know microtubules, they serve as the passageway or the highways of the cell. So they allow the microtubule, the organelles, to move within the cell in a particular direction using energy from ATP. So with reference to figure 5.2, explain how the enzyme that are secreted by the cells of pancreas are packaged into vesicles and exported. You have to describe packaging, and you have to give a description, explain the packaging of vesicles as well as exportation. So enzymes produced when rough ER, they are transported to Golgi. The cisternae of rough ER delivered these to Golgi body. We know rough ER is the one synthesizing enzyme because enzymes are proteins. They move to Golgi for modification. The enzymes and then packaged into vesicles and sorted out. The Golgi vesicles carrying enzymes move through the cytoplasm along the microtubules using energy from ATP breakdown. The vesicles is having phospholipid bilayer that fuses with the phospholipids of the cell membrane. The enzymes are then released out of the cell by exocytosis. The last part is a bit tricky because here they actually take you to water and they ask you to say that you have to state the property of water that allows for each of the following to take place. If we say you have to be specific with the property here, nothing else they did put anything any description here, just the property. The solvent for glucose and ion, which property of water allow it to be a solvent for the glucose and ion? Water is a polar molecule, so polarity is the property. Move it up with the xylem. So yeah, it's hydrogen bonding, dapple interaction, cohesion. You can mention that's the property of water. Helps to decrease temperature in body of mammals. So latent heat of vaporization is the property which we can mention in this at this point. This question came in October-November 2016. This is variant 21. This enzyme lysozyme is shown to you. This enzyme lysozyme, these are all the amino acids which are joined together in the primary structure, one after the other by peptide bond. And as you can see, the chain starts to fold up. Why it's getting folded up? Because of the secondary and the tertiary coilings. And this is just a single polypeptide, so it will not have a quaternary structure. So these are the disulfide bonds. The disulfide bonds will be there between two cysteine amino acids because we know that's the only place where disulfide bond is formed. Disulfide bond is a type of R group interaction, and among R group interactions, this disulfide bond is the strongest. We know it's the strongest, and why it is the strongest? Because it is a pure covalent bond. The question now describe name two type of covalent bonds shown in the structure, the figure 3.1. So that's disulfide and peptide bond. The primary structure of lysozyme. Explain the meaning of primary structure. The primary structure is always the sequence or the order in which the amino acids are joined together via peptide bond in a protein. Lysozyme hydrolyzes one beta 1,4 glycosidic bonds present in the compound found in bacterial cell wall. State what is meant by the term hydrolysis. So hydrolysis is always the splitting of a larger molecule into its monomer using enzyme and water. So both need to be there for it to be broken down. So there's a type of biological molecule which will be the substrate for lysozyme. Yes, a carbohydrate can be there because it's breaking the glycosidic bond. So the type of biological molecule will be carbohydrate. Lysozyme uses induced fit mechanism. Explain the mode of action by using the enzyme that use induced fit mechanism. So we know the induced fit mechanism, the enzyme is not exactly having the active site, which is complementary to the shape of the substrate. When the enzyme holds the substrate, it molds its active site slightly to accommodate the substrate for a better fit, and ES complex then start to form. In human tear cells, saliva, lysozyme acts as extracellular enzyme. Why is it extracellular? Because the enzyme is working outside the cell. This figure shows you investigation of effect of substrate concentration on the rate of catalyzed reaction by lysozyme. You have to state the lowest substrate concentration that would give the Vmax. So we know Vmax is when the graph becomes parallel to the x-axis. So if you look closely, the graph becomes parallel to x-axis, and this is the lowest value, which actually led to the Vmax being there. So we'll just look for this point, and this point actually turns out to be 2.7. So 2.7, we need to mention the unit with it. So that's 2.7 millimole, lowest substrate concentration that gave Vmax. Then they're asking for Michaelis-Menten constant. The Michaelis-Menten constant, how that will be calculated. So for that, we need to just find out what's the Vmax here. So the Vmax, we'll just look at it here. Look at the graph. We'll just look for the point which is pointing here. We'll read it off from the graph what this is. So this is 3.2. So when this is 3.2, Vmax is 3.2. What will be half Vmax? So half Vmax is half of 3.2, which is 1.6. What I'll do, I'll just try to look where 1.6 is on the y-axis. So the point where 1.6 is present, I'll just draw a line here. I'll touch it on my graph and I'll drop it down on the x-axis. The value on the x-axis will be my Km, and Km is a substrate concentration required to achieve 50% saturation of the enzyme. So according to the definition, the value of Km is 1 millimole here. So we write down as 1 millimole. The investigation was repeated in the presence of a competitive inhibitor of the lysozyme. Draw a curve to show expected results. So this is how the expected results would be, because when the competitive inhibitor is there, the rate of reaction will start to slow down. But eventually, as the substrate concentration is increased, gradually the rate of reaction will start to increase, and eventually it will reach the same Vmax as it was without using the inhibitor. If they were asking you for a non-competitive inhibitor, the graph would go somewhat like this, because it would not go to the same Vmax as it was going before. This question of variant 2015, variant 21, it shows to you how the enzyme interact with the substrate. So you can see the enzyme and the substrate here being bound together, and the substrate is an exact fit of the enzyme's active site. So that's this enzyme is working by lock and key. Whereas if you look at the other enzyme, this enzyme B, the active site is not exactly a perfect fit for the substrate. So this enzyme is not working by lock and key. This is working by induced fit. So explain the difference between the two ways in which the enzyme interact with the substrate. So you will just highlight how the lock and key example, how lock and key works. The locks and key, they fit into each other because they are having a complementary shape. So they don't bring about any change in the shape of the active site anyhow, because the ES complex forms just by fitting of the substrate into the active site, because they are an exact fit. They are complementary in shape. Whereas the enzyme that work by induced fit, the enzyme is having an active site which is not exactly complementary in shape to the substrate, but it allows the substrate to come. When the substrate comes in, it molds its shape slightly. It changes its shape slightly. This is because of this shape change is induced because of the substrate fitting into the active site. So that's what you'll be highlighting here. They are mentioning carbonic anhydrase is an enzyme found in blood and liver and the kidney. Show a molecular structure of shows a molecular model of this enzyme. So with reference to the structure parts labeled P and Q, explain the term secondary structure. So in this, you can see the arrangement. These are the beta sheets. So beta sheets always look like these strands. So you can see that they have mentioned arrow as well. So you can see one is going up above, the other is coming down, then one is coming down, the other is going up, something like this. So these are all beta sheets. So P are beta sheets here, whereas Q, these are all alpha helices. So you can see alpha helices are always like spiral staircase, and they are like turns around that, which are spring like that. So you will just mention how it shows, and we know that in this, because why are you having the secondary coiling? The secondary coiling is because of the hydrogen bond between the carbonyl, the carboxyl group, and the NH terminal of the peptide bond, because they are having a slight negative and a slight positive charge, so they form a hydrogen bond, and both these, they belong to the peptide bonds of different amino acids. So that's how the secondary folding takes place. Describe the role of carbonic anhydrase and the enzyme. We know that when H2CO3 is present, it dissociates in the presence of the enzyme carbonic anhydrase into CO2 and water, and this reaction occurs in lungs. And when it occurs in lungs, it allows carbon dioxide and water to go out and exhale. It is exhaled, it goes into the alveoli, and it is exhaled out. The hemoglobin is then available back for binding to oxygen. So that's the function of the enzyme carbonic anhydrase in blood. This question is from Agent 2018, the variant 21. They have shown to us the structure of hemoglobin molecule, and hemoglobin molecule is shown. They are saying it's the rapid model of hemoglobin molecule, and this is structure. You can see these are some coils here, and again you can see these are some random loops here, and you can see these structures of alpha and beta globin. These strands at the back, these turns, these are the beta ones at the back. This is an arrow. You can see this is the beta turn as well. So beta turn will always have arrows, and these arrows actually shows to you that this beta is going in that direction. So an alpha and beta, then they are joined together. So what structures which are having, we are having certain letters, we have to identify them. So a chemical group that is not made of amino acids, it must be a prosthetic group or a heme group. Form of secondary structure that is found polypeptides in a hemoglobin, and this is a spiral structure that must be alpha helix. Each polypeptide is folded to form complex 3D shape. That must be tertiary structure, because tertiary structure is always a single polypeptide folding it into a 3D shape. Primary structure is when a polypeptide is made from a sequence of amino acids joined together. Quaternary is when multiple polypeptides are joined together, as shown by the shading of this molecule. That's it from my side. I hope that was helpful. Thank you so much.